Hello, my name is Dr. Gordon Chu, and you're hearing my voice go through the slide deck for Phenegra's innovation in plant nutrition and crop development, seed to seedling discovery. Imagine if we could do this in a healthier, more efficient way and in half the time. You see all those rows of lettuce? Imagine that. Imagine understanding the plant microbiome to a point where the phyllosphere, the endosphere, and the rhizosphere is understood, particularly at the rhizosphere, where you have the zones of influence of root secretions, exudates, and soil microorganisms working together to allow you to have a healthy endosphere. That's where you have your internal plant compartments, as well as your aerial plant surfaces, the phyllosphere. That can only be achieved at a healthy level if the rhizosphere is healthy. So in slide 4, you see under the healthy rhizosphere what is necessary. It is understanding that the seed microbiome needs the microbiome. It needs the healthy soil as well as a healthy ecosystem so that you can have the seedling form from the seed. In slide 5, the plant roots and diverse soil microbes exist in the rhizosphere microbiome. Basically, the roots environment requires the plant root with a diverse soil microbe in there that you might not be able to see. Here in this illustration, I've made it possible for us to visually see those soil microbes. Healthy soil microbes consist of type 1, 2, and 3. So they work together. So you might see some overlap, but they all do some overlapping sweet spots as well as additional things. Type 1 in slide 6 says inhibits growth of bad guys, pathogenic bacteria. That's good. Inhibit growth. You don't want the bad guys to grow exponentially faster than the good guys. It facilitates the breakdown of organic matter. Well, that's important. And then makes nutrients more accessible to the plants. Also quite important. Enter type 2. Type 2 soil microbes aids in decomposition of organic matter. Oh, so isn't that the same as facilitates breakdown? Yeah, somewhat, but I changed the words a little bit to highlight that they also do a overlapping job. And then they have decomposition releasing vital nutrients. A little bit more than just the decomposition side is releasing those vital nutrients and the nutrients enhancing of the soil fertility. Okay, then you have type 3. Oh, you have complex carbohydrates. I can do that. Type 1 and type 2 doesn't have as much of an ability. It also improves the soil structure. So then you don't have as much compaction. It also improves nutrient availability. Oh, well, that's type 2. And it also is type 1, but that's where you have some of the overlap. Nevertheless, type 1, type 2s, and type 3s in our system is not just 1 or 2 or 3 of these, it's a complexity of them. So, what could possibly go wrong in nature, right? Or today, what's going on that's wrong? In slide 8, we see that we have a depleted and undernourished soil system. We have biological depletions. We have decreased microbial activity. We've got pathogenic threats. We also have chemical situation going on where we have usage of synthetic fertilizers. Why? Because it wasn't working naturally. Oh, possibly with the biologics, but also, we threw in some pesticides. Why? Because they're attacking our food. So anything that attacks our food, we got to kill them. So what have we done? We had to use pesticides, insecticides. Nobody wants to eat food with holes in it, right? But that also leads to pH imbalances in what we are doing in order to achieve the end game, the goal. Ecological challenges are we now have environmental stressors, more so than before. We have environmental extremes. We've got erosion issues. We've got disrupted ecosystems. And then physical, intense tillage and compaction, right? So that combined, these 4 issues are leading to undernourished soil and depletion. Seedling disease and pests are another area that we are having issues with. We've got pathogens, fungi, water mold, known as mites. We've got bacteria and viruses. Those are just rapidly hitting us, as we can see in slide 9, and we get root rot. We get seedlings that don't make it. We get seedlings that are impaired. We also have the other system. These are the ones that you can see. Those are the insects and the protozoas. So what does a farmer do? Throw in more seeds, right? But then when the seeds turn to seedlings, the ones that make it, they don't make it well. So it's like it's compromised. And then now you still have to feed your seedlings post germination stage and grow it into plants. And then you have to treat the plants because now they're sick plants that you maintained and allowed, but could have addressed during the seedling stage. So let's look at what we have also done. We have used antibiotics, and the antibiotics have now created a target for the harmful, but also the beneficial microbes. So now we continue to lose type 1, 2, and 3s. We've disrupted the natural soil microbiome because of the philosophical approach of trying to address the problem, bandage after bandage, led us to this big problem, right? We just have to use more and more chemicals, more nutrients, rather than addressing the source of the problem. We have disturbed the plant absorption and nutrient utilization. We have caused the decline in soil health and fertility. Well, what's considered to be an environmental pollutant? It's antibiotics. And that's an environmental pollutant to your soil, to your water, to your sediments, and to your farmland. One might even consider farming to be mining, right? When you're growing the crops, you're mining the soil of these nutrients. And then you're saying, oh, let me just put some NP and K, nitrogen, phosphorus, and potassium back in. Let me rotate my crops. But you can see that has not led to a decrease in the usage of pesticides and insecticides. It's only led to more potent systems. We've even had to modify the genetics of plants so that we can keep our food levels up. We have led to incredible detriments to the nutrient cycle in soil from the advent of antibiotics. So in slide 11, soil nutrition and human health. Soil nutrition, which is critical for plant nutrition is critical for human health. Well, this wonderful cycle is now disturbed. And when we have undernourishment of food, right, we then have food insecurity. So how do we end up getting this undernourishment? We get lower crop yields, poorer crop yields, poorer nutrition, quality of food. We have a compromised human physiological function that results from this. Our children and adults end up having weakened immune systems. So we have disease. You might think back at COVID-19 to other outbreaks. Why is all this happening? Are the bugs really stronger, or are we gotten weaker? We end up eating, but we end up having reduced energy levels. Impaired cognitive function is widely prevalent in our children as well as in our adults. Who's a vulnerable population with a heightened need for quality soil nutrition and end up can't address this? Well, it's the elderly, the sick, the infirmed, the wounded. You have wounds, you need quality nutrients. If you're impoverished, if you're an athlete, those sets of populations also need that. And financially insecure, yes, you want to be able to get your nutrients. Otherwise, you're going backwards. So enter slide 12, Phenegra's innovation, which is the PLPS. What is it? Well, the liquid, the pod, and the seed. So if we look at the liquid, it's a proprietary formulation of the type 1s, 2s, and 3 microbes. This leads to extraordinary nutritional availability, probiotic synthesis. The pod, basically the structure for the germination, this is a novel nutrient-rich seed-to-seedling ecosystem. It contains intensified microbiomes because it's been treated using a proprietary system to get that ecosystem completely nourished, right? We use temperature control to put it in there. That leads to increased seed germination rates and yields as we go through the slides. You'll see that. And then it leads to prolific seedling and root structure growth. That will be very evident in the slides that come. Under Phenegra seeds, the PS system, right? PLPP and then PS, PLPS, the seed needs to be treated with the liquid under a proprietary ecosystem and approach, right? This promotes the seed germination. It protects the seed from the disease. So you don't just put the seed into the pod before treating it, right? You want all your seeds to be treated with the P liquid, but it's not just the liquid, right? You also need it to be treated under a certain scientific controlled environment, right? That methodology is important. But obviously you don't need to only rely on that, because if you only relied on P seeds, you wouldn't need P pod. Well, the pod gives you a radius, three-dimensional radii of basically a barrier, like a force field around it. And then as there's so many rains and other ecosystem attacks that the seedling will experience, you need to continue to add some P liquid into your ecosystem, right? But the heart of it is really PLPS. Right. So now we go to slide 13. Here is an actual Ferrari bush bean seed grown using the Phenegra system. Now, treated with PLPS versus untreated is what you're looking at right there. On the left, you have the treated system. On the right, you have an untreated system, which leads to the mold and fungi setting in. Treating prevents the mold and fungi from setting in and leads you to having greater success rates. So on a controlled in a controlled experiment, you had the same room, identical containers, you had the same ambient light, the same ambient temperature, same drip irrigation, that's what was used here, and then the same growing time frame. What resulted was a faster, healthier seedling using Phenegra's PLPS. The other seeds never made it. They never got to germinate. They were attacked and taken out by mold and fungi. That makes sense. Slide 14. Here is an actual watermelon plant grown by Phenegra. Again, using the P liquid, P pod, and P seed. Of these four pictures on the very left, you have the P pod seed to seedling rhizosphere substrate. To the right, you have prolific root growth happening. Then you have the third picture where you have prolific dendrite surface area for water and nutrient uptake. Look at those dendrites forming off of the roots. That increases your capabilities. And then on the very last picture, you have a healthy, nutrient-rich watermelon plant. So this forest from trees approach, right? The forest is, oh, I see all my plants, but the roots tell you a lot. And that's going even deeper than the trees. Forest and trees is, oh, okay, we need to go to the roots and see what is going on when we have type 1, 2, 3 microbes helping out. All right, so now on slide 15, we have other plants, lettuce, chard, peppers, and tomatoes as actual produce grown by Phenegra. On slide 16, we have the actual root systems. Look at the dendritic offshoots from the root structures of these seedling stages. These are not like massive plants, adult plants, but they're at the seedling stage, and you have this huge ball of roots with lots of dendrites, high surface area. Guess what's going to happen now as they grow into the adult stage, right? They're just going to pick up lots of nutrients. So The applications are very wide. They're agnostic. It can help with indoor, outdoor, as well as scale production. Indoor, you got hydroponics, you got soil-based systems, vertical, as well as controlled growth environments. That can all benefit, right? Who can't benefit from having the roots have extra dendrites, right? So then you have your outdoor. Well, the outdoor is the Wild West, right? You really don't know what's happening at night, what's going on during the day. You can't monitor it. You have all kinds of weather conditions, and just the beasts are out, right? At least in the indoor system, you have some level of control. Outdoor, there's no control. It's just the Wild West with the sun. Sometimes there's the rain, but there's lots of bugs, and there's other microbes that are attacking your system in a highly infected environment because we have antibiotic usage and it's everywhere in the soil. Um, this really impacts at scale, right? You've got industrial agriculture that needs it, commercial, urban environments, your local grower, you as an individual consumer and grower, you're going to grow all kinds of things and you want them to be healthier. Well, what is healthier? And I'm showing you getting attacked doesn't lead to you being healthier. Um, and then once you have an infected seedling, you don't know. So or a seedling that doesn't have good root structure, you're not going to know that either. Most pots are not transparent. So you're just going to feed everything. You buy all your fertilizers, you put more fertilizer in, you wonder why is it not growing well? Well, you can't see and you don't know, but you still have to feed them. That's a cost. So in slide 18, the advantage of the Phenegra LPS system is the rhizosphere modification. I think that becomes really apparent by this point, is the modulation of your microbial community at the start, at stage 0. Beneficial changes in your soil structure come from stage 0 into seedling stage. Well, those beneficial changes in soil structure came from type 3, and type 3 works with type 2 and type 1, so that you now have improved nutrient cycling. You have enhanced plant microbe interactions, and you have positive influence on the seedling establishment and growth. Basically, the soil, if you're using soil, right, the substrate is now going to be healthier. And for reuse, it has less compaction issues. The rhizosphere protection is unique to the Phenegra formulation of using type 2, 3, and 1s. The advantage is here in slide 19, you end up having a biofilm formation on the seed surfaces. This is the good stuff, the protective microenvironment that we just don't see. Enhanced seed hydration, well, that's really important. You want your seeds to be hydrated, but you don't want mold to set in. So how do you protect that? That is using the microbes. You end up enhancing your nutrient uptake. We know that now. We end up having resistance to drying out, basically. That's desiccation. And then what happens? The result is improved seed germination, facilitated water absorption, physical barriers against things that you can't see, pathogens you can't see. And then environmental stressors, yes, we can feel those. We can even measure them. But oftentimes we're so busy, we don't even know it's happening and it's too late. So better tolerance of that is crucial. Better tolerance of your ecosystem, because your ecosystem is no longer as reliable when it's outdoors. So here we are, here we have the biological seed stimulation side. Probiotic microorganisms at the heart enhances germination. These are known facts, right? We have induced systemic resistance, ISR. We trigger the plant's immune response to be optimized. We increase its resistance to pests as a consequence, because if it's optimized, the pests will say, oh, I'm not eating that. That's too much to eat. So in this picture with the cabbage that had a better seedling stage experience, well, that's going to now be able to grow stronger. And oh, I'm not going for that one. I'm going for the other sicker ones, right? It reduces vulnerability to infestations because pests lead to more pests. So if they're eating, they're going to produce more pests, and then they're just going to attack more plants. If your plants are healthier, you're not going to harbor as many of these nasty pests in your garden. And then your beneficial bacteria, as well as your beneficial insects, are going to work together to give you a great growing season. Um, Microbial antagonism is a very important concept, is that the ecosystem at the seedling stage helps repel the pests. It's a natural form of pest control, right? Because pests go for sick plants. But then after they feed on them, they do what pests do, which is make more pests. So nutrient availability here in slide 21 is a critical understanding of probiotic microorganisms help solubilize nutrients. Enhancing that nutrient accessibility to plant roots is what you need because that leads to essential elements for seed germination. Essential elements are necessary. It's like good practice for early seedling growth. By promoting the vigor and the establishment of the plant in that substrate, you now have improved plant uptake of the nutrients. Now, nutrient retention and delivery at the improved soil fertility and nutrient cycling level leads to a uniform nutrient absorption. And then you end up reducing nutrient leaching. Hormonal regulation in slide 22, probiotics microorganisms produce growth promoting hormones. Well, these hormones regulate plant growth and development. They stimulate seed germination. It stimulates seed dormancy breaking through. You have enhanced root elongation and overall seedling vigor. Well, what happens if you don't have the treatment, with so much antibiotics out there, you end up having the opposite of this. And so oftentimes you say, oh, this is such a weak plant. Why is it not growing strong? The answer lies in the roots. And underneath the roots is what's really going on when you see slide 22, where we pencil in the chemicals. Well, now you can see the chemicals. And those are the nutrients. Those are the components, but you can't see that. All you see is, oh, my roots may not have as many dendrites. Huh. Well, but now the plant is grown to a point where it looks like it's okay. Well, now you have to feed it. So you got to buy and spend a lot more on fertilizers. So soil pH regulation is also crucial, right? When you have the right microbes, it selectively absorbs and releases hydrogen ions. It achieves and maintains optimal pH levels. Well, if you don't have those microbes in there, well, now you're going to be subject to more fluctuations. But the key really is you're like raising all these zombie plants, and the plants aren't healthy and they're somewhat sick, but you now have to still feed them, and they have a good appetite. It's just that imagine all that wastage, right? And then you're now getting plants that are not great. When you are ready to source your food, or imagine growing flowers like roses and things like that, it all cascades down to your pocketbook, but also to your health and to your overall wellness. Improve soil structure and root health. Slide 23, reducing compaction, right? Otherwise, you have to reduce the compaction mechanically, or you have to reduce the compaction monetarily. You have to buy things that reduce compaction. We end up having stable aggregates. We have a more porous soil structure through the roots with more dendrites. Because think about what roots do. They break up the soil. You have improved water infiltration because of the broken through soil, because of the improved root penetration. And then root penetration, what is it trying to do? It's trying to breathe. So you have enhanced air exchange within the soil, and you end up having improved nutrient uptake. Water retention and conservation. Isn't that what we need to do? So having more root developed structures, advanced structures, we improve our water uptake and retention. We reduce our irrigation requirements. And if it's uptaking better, we also improve our runoff, right? Now, in slide 24, the enhanced stress tolerance is really important because we do live in an environment with crazy temperature extremes. And just as an example on slide 24, you got the cracked soil from drought. All right. You've got salinity. Well, because we some environments have snow and we don't like snow, so we want to melt it faster. So we're using various salts on the roads. And whether we like it or not, we are experiencing extreme weather conditions, temperature extremes. Well, better roots, better tolerance, more dendrites on the root, better tolerance. How do you even do that, right? But now you can do that. You can mitigate the effects of environmental stress on seed and seedlings. You might not be able to control the weather, but you've improved your response to it by giving your plants a better treatment at the seed to seedling stage. Actually, there was no treatment for the seed to seedling stage. You just basically said, oh, germinate, and then we'll deal with it. Now we're changing that. Disease suppression. Probiotics antagonize and compete for resources with plant pathogens. So, what that means is the good guys fight and compete for the same resources as the bad guys. So, more good guys, nothing left for the bad guys. Probiotics, the good guys, protect, therefore, your seeds and seedlings from infections because the bad guys weaken the plant, weaken the seedling. It might not be attacking the seedling. Sometimes some bad guys do, but not all bad guys do that. But it weakens its ability, and consequently, other things like the mold, like the single cell organisms, or like multi-cell organisms such as insects, right? So what are the advantages, right, in slide 25? The resource efficiency is there. The reduced environmental impact is there. The optimized nutrient and water management is there. Root growth improvements. There's nothing more obvious than the dendrites on those roots. And then you end up having enhanced disease resistance. The plants are healthier. So they're just going to have more momentum for them to grow. They're going to now absorb the nutrients better, as opposed to you pouring more nutrients from your pocketbook into the soil. Precision monitoring and growth of controlling the conditions at the seed to seedling stage is like sending your kids to the ultimate mindfulness mindset change at the preschool kindergarten level. Yeah, that was more expensive to do that and to treat it because we originally did nothing, but now we don't have to go and worry about them because now they have a good path forward, right? The path is maintained. This is repeatable. Generations will be able to benefit from this knowledge that we have found in our breakthrough. So in slide 26, Phenegra's seed to seedling innovation at scale overcomes existential threats to humanity, the preservation of our natural resources, the abundant and sustainable crops, the nutrient rich foods that we'll get, the vitalized natural ecosystems, and even revitalization, right? Not just vitalization. Natural disease resistance, crucial because, like I described earlier, when you have disease, leads to more disease. Pests that eat your plants make more pests. So now we just have more pests, and now we have to use a lot more insecticides. Otherwise, if we don't do it, we're gonna blow up the insects because they're just nasty. They're having all this. Well, we led to them because we gave them weak plants, and because we couldn't detect it, we now have food security issues. So elimination of food insecurity, as well as reduction of environmental pollutants, was we don't have to use weapons like that. All right. So as a bigger picture, when you buy and support Phenegra, you should look at where Phenegra came from. The gra in Phenegra is really graphene. And my initial breakthrough discovery that led me to having the ability to be the scientist that you know is the creation of transportable reactive graphene. And I once said that it will prove to be one of the most consequential discoveries of our time, advancing new material science research and innovations for decades to come. At Harvard University, Phenegra won the Harvard Tough Technology Award in 2023 for graphene innovations within asphalt. One of the big subsequent explosions was achieving the Wharton Award, as well as then being selected for the economic development in New Jersey in 2024. So what you're supporting when you buy this is a fragment, and it's a big secret. We don't want to give away secrets, but this particular approach to making the asphalt better involved the graphene, the transportable reactive graphene, but it also involves some very, very special components in the secret sauce that also do other things. So the system you're looking at has no graphene in there, but it uses the microbes. And when we can put microbes into helping us grow better, it's an extension of the Phenegra philosophy. The DNA of the company, of the startup, is what if one day, through our advances and through our intentions, um, we can create not only better food and better crop, but what if we could one day make asphalt arable? Not the asphalt that we drive on right now. That's toxic, right? But imagine if we could one day come up with something totally different that we drive on, and when we're done driving on, it actually can be used as a dual purpose. Well, that's not what we have right now, but what this is what we want. And I believe if you agree with today's presentation, it's what we need. We need and we want to have a better outcome for our children, for ourselves, for society, without using bandages, without having to hurdle over such high expectations of, oh, let's blow up more insects. Let's just have more zombie plants. Um, and and then just having to feed them more. We're wasting. Change that mindset, change the future. Thank you for supporting Phenegra.